it's kind of funny. I'm going to be talking a little bit about CAR T cells. It's probably the first time a CAR salesman has come to exponential medicine. But what I'm going to be talking about is an evolution that's happened over 30 years in this space for trying to use T cells, one of the body's defense mechanisms against cancer infection, to modify it to treat cancer. So what I'm going to do is just walk you really quickly through the history. This has been evolving for 30 years. So back in the 80s, out of the NCI, there was a foundational discovery made for a growth factor called interleukin-2 that allowed you to grow T cells in culture. And so what happened was scientists took T cells out of a cancer patient's body, grew them to large numbers, hoping they would treat the cancer and put them back into the patient. Well, unfortunately, while there were signs of some clinical effect, it didn't work, but it really spawned the evolution of the rest of this field. And with the evolution and development of new technologies, things for selecting cells, for growing different types of cells, as well as evolution of things like gene modification, viral vectors, there was a whole slew of different T cell therapies that have been evolving for 30 years. And what I'm going to talk about today is the most recent success story, which is the chimeric antigen receptor gene modified T cell. So what is a CAR, besides the thing you drive? Basically, it's a genetically engineered receptor where we take components from different cell types. We take the antigen binding region from an antibody that is specific for a tumor antigen. Very high affinity, it'll track down, bind to a tumor with very high binding capacity. We weld that into some of the elements of a T cell receptor. So a T cell receptor is on the surface of the T cell. This is the thing that sees virally infected cells or other cells that need to be eradicated. It sends signals through this signaling complex into the cell to kill the target cell, to proliferate, to make more of itself, to find additional cells with that same antigen, kill them. So we've welded these two pieces together genetically, and then we add another component which we call a co-stimulatory domain. It's another receptor on a cell that has elements that tell the cell to do certain functions. So if you want to use the car analogy, this could be like the GPS system here that tells you where to go. This could be the gas pedal, turns the car on and off, and this could be the steering wheel, tells you where to go and how to steer and how to get there. So that's what I envision the new car is, but it's actually not that sexy. It really looks something more like this if you look in the literature, not through a microscope, but it does have this domain from the antibody that binds to the tumor antigen very specifically, has a transmembrane region that anchors this in the membrane of a T cell, and then has these signaling domains we've engineered into here that will tell the cell to kill the target that it's bound to, cause the cell proliferation, recruit other members of the immune response armamentarium. So I'll give a little video here. This is, or I hope this, well actually, what's underneath this, I think we tried to modify it so this would play. This is a little video, but underneath it what it shows is that you take a patient's blood cells, you purify the T cells out of the blood, you genetically engineer them with a vector. It could be a lentiviral vector, it could be CRISPR gene editing, to make every T cell that you modified specific for the tumor, for the cancer antigen. And then you grow them outside the body to numbers that we think are therapeutic, put them back in the patient. And I don't think this video will work, but what it shows is there's a bunch of little T cells in here and a bunch of tumor cells. And if the video, it might work. There we go. They will recognize the tumor, they'll bind to it, they will kill it, they receive a signal to proliferate, creating more clones of themselves. They'll go out seeking new tumor, and they'll continue this until the tumor is eradicated. So, like I said, we've been working on this for 30 years, and I can just say that back in about 2011, 2012, when I heard about the first 3 patients that Carl June treated at UPenn for CLL, and all 3 of them went into complete remission. And, in 30 years in this space, we get hints of activity, a few patients who respond. This was profound, and literally, when I heard him talk about this, I had tears in my eyes, because I realized that we've been working on this, so many people working on this that it's finally working. And about the same time, out of Memorial Sloan Kettering, out of the NCI, similar protocols move forward with the same results. So this was in 2012, so in less than 5 years from these phase 1 clinical trials, we now have 2 approved, the first in the US, gene-modified T-cell therapies for cancer. And they're working so profoundly that even the guys at the FDA, kind of like me, I don't know if they had tears in their eyes, but it's pretty exciting that we've developed a living drug that really, really seems to work. So there are issues associated with a living drug. It's not a small molecule, it's not a recombinant protein or an antibody that you can make in E. coli in large vats. And so one of the issues with making a drug from your own cells is that every patient is different. Not only are they different genetically and have different backgrounds, but their cells, the time we collect them could be different. They could have had a chemotherapy, they could have had a stem cell transplant, they could be old, they could be young. So there's a lot of variability already built in there. And then there's also when we manufacture the cells outside the body, it's missing all the cues the body would give it. So we're doing our best to grow these, maintain them in their potency and their ability to persist and be safe. So those are all issues that we all have to address, but probably most important to me is in a living drug, how do you really understand what you're dosing with? Sure, it's a cell, but they all have different properties, just like we all have different properties. And how do you make sure that you dose appropriately while reducing toxicity? And I think all these little clips from various news things show that, yes, there's toxicity, there's neurotoxicity, there's cytokine release syndrome, and there's been a few deaths, but the FDA has approved these, so the benefit for patients who have failed all prior therapies in the setting of leukemia, and in the case some cases of lymphoma here, where we're seeing 80-90% complete response rates, and a lot of those are durable. It's starting to look like a cure. I think when we're 10 years out, we'll feel more comfortable. But we really have to, even though we're saving a lot of patients, we don't want to lose any because of toxicity because we don't understand our drug deep enough. So what I'm going to talk about here just for the remainder of the talk is the tools we use for deeper understanding. There's a classic toolbox that people use, like flow cytometry, ELISAs, functional assays, mouse models, and looking at RNA expression that people have typically used over the years to characterize and identify their products. But now we're going deeper. We're using very advanced technologies, using mass specs, CyTOF, single-cell sequencing of the genome. I'll talk a little bit today about one method, ATAC-seq, which we'll come to in a moment. And then we're taking all this data. We have tons of data from not only the blood we get from a patient, but as we make the drug from their own cells, the drug that we're about to fuse in there. And then we can also collect blood from the patient, biopsies, and really understand for the patients who are responding, not responding, having toxicity, take all that big data and now apply the tools for analyzing it and understanding what's causative and what we can do to make the drug better. So I'm just going to spend like 1 minute on ATAC-seq. I love it because it's a new technology that was developed in 2013. And what's really cool about it is it's just a tool for actually looking at your chromatin, your DNA, your genes. What's open and available to be expressed, what's open and ready to be expressed, or what's open and being expressed, or what's closed and it can't be expressed. It's kind of like wiring, like how will this cell respond? What's its potential? It's kind of like looking into the future. At least for me, that's what it feels like. And so this is my data slide. The only point I'm going to show you is that it's kind of cool because you can see along anywhere in the genome all the genes that are open. And you can actually see the control elements that control their expression. You can see if they're occupied and being expressed or if they're not occupied and just poised to be expressed. So it's just a very fascinating, revealing kind of wiring of the cell. So I just want to show a couple pieces of, this is from a clinical trial that we were doing in the setting of leukemia where we applied this technology. And what I'll say is we use computer algorithms to look at the whole open chromatin status of cells in our drug product that we're giving to patients. And then we correlated that to their clinical response. And we have a few what I call normal donor drug products that are comparators. When we clustered these based on similarity, we found that this cluster, all the normal donors and all the patients who were complete responders clustered together, whereas the patients who had progressive disease or partial responders clustered together. So if we dig down even deeper and look at individual genes or gene attributes that might reflect inflammation or senescence or mobility, we can start to pick out genes, and of course our IP council would not let us reveal what those are publicly, but we can drill down to specific genes, and not every gene correlates with response, but we're finding many, many, many that appear to correlate. Numbers are small, we're continuing this, so it's all with caution, but it's looking very promising. So what can we do with all this information that we have? We can look at the cell surface proteins, intracellular proteins. These are kind of the remnant of translation from transcription from open chromatin. So I actually look at this whole process here, protein, RNA, chromatin state, as kind of a trajectory of the cell. The chromatin state's the cell potential. What can it do? RNA is kind of the downstream from the open chromatin, and then proteins even downstream from that. And so what can you do when you understand the trajectory of your drug product? Well, the hope is, instead of taking a snapshot in time where you get a little piece of data and you say, oh, we have 9 or 10 spheres here, we have information about them. Once you know their trajectory, you can actually tell a lot more about them. You can say how they relate to each other, their interaction, and you can predict where they're going to go with tremendous accuracy. And I think that's where we're trying to go in this space. So what we're doing is we're leveraging big data. We're collecting everything: RNA transcriptome, signaling pathways, patient response, clinical response, state of the chromatin, gene expression, and we are consolidating it, and we will be moving that into what we call a funnel, where we take all the data along with clinical data and then put it through some of these analytic tools, such as machine learning, that allow us to extract what we think are really strong correlative markers for good or bad. And then we can start engineering ourselves better to accomplish better response rates and better safety. So the goal is, this is the only conference I could ever go to and show tricorder and probably not getting people to laugh. But the goal is really, can we take this and define a limited set of markers we can use very inexpensively, very reproducibly, to continually monitor our production of new drugs and identify from a drop of a patient's blood before we manufacture their cells in an expensive process, can we identify if they'll even make a product that they can respond to? So if we know they can't by these markers, we don't make an expensive drug product. And that way we can start treating only people who will benefit, and we can understand better the dose. How much can we give and get the best response and yet avoid toxicity? So I think I'm like a minute over, but I thank you very much.